Welcome to the molecular evolution session. The first talk of the session is by Chris Hittinger, so it's a change of the speaker, please.
Thanks, Dimitri. So as he said, those of you who are skilled at phenotyping vertebrates as opposed to fly or yeast will realize that I'm not Meihua Kuang, but rather Chris Hittinger. I don't know whether, are there slides, perhaps? Oh, there we go. So here is Christina again, some phenotypic differences. Christina really wanted to be here to present her own work, but her father passed away suddenly, but she really wanted to make sure that we got a chance to share this exciting story with you. And so with the caveat that advisors seldom do as good of a job of presenting their students' work as their students would, we're going to dive in and tell you a little bit about ongoing resolution of duplicate gene functions and how that has shaped the diversification of a model metabolic network. And so there's, of course, been a lot of thinking and a lot of study and theory on gene duplication and its contribution to evolutionary novelty. And I would argue that really a lot of the attention has gone on the process of fixation and how genes have to differentiate themselves either through neutral mechanisms or through adaptive mechanisms so that both copies can be maintained. But of course, after that happens, you could have hundreds of millions of years where those duplicate genes can undergo continuous divergence. And of course, this can result in differences that exist between species. And so the potential actually to continue to have duplicate gene differentiation impacting phenotype is quite profound. So the model system I'm going to tell you about is the classic GALactose utilization network from Saccharomyces cerevisiae and its close relatives, and it's been worked out in exquisite detail over the past several decades. So this is an alternative, non-preferred sugar. Saccharomyces would much rather use glucose if it were available, and as a result, it has a very sophisticated system to sense galactose. And so there's the transcriptional activator GAL4 is actually bound by the co-repressor GAL80 that keeps the network off when galactose is not there. When galactose is present, the co-inducer GAL3 binds to galactose and also to GAL80, pulls them off of GAL4, and then allows GAL4 to activate the entire gene network. The gene network comes on, and it encodes the transporters and enzymes necessary to bring galactose into the cell and perform the necessary modifications that it can go through glycolysis and then ultimately either be fermented or respired. So it turns out you don't have to go very far from Saccharomyces cerevisiae to get some really profound differences in gene content. And so we know there's another yeast about 100 million years divergent, Chloromyces lactis, that did not undergo whole genome duplication. And so we can see here it has a single co-repressor and also a gene that is bifunctional that performs both the first enzymatic step and the co-induction function. And the entire network was duplicated about 100 million years ago, and the genus Saccharomyces originated about 10 million years ago. And in the last 10 million years, there's been differential retention of duplicate genes. And in particular, you can see the network in cerevisiae is a bit more simplified. Indeed, in one population, it's been entirely lost. And in this talk, we're going to focus here at about human chicken level sequence divergence and study what the function is of the second copy of GAL80 as well as the copy of the co-inducer galactokinase. And to cut a long story short, I'm going to show you the data, but to give you the answer up front, what I'm going to show you is that the network is actually more active and the gene duplicates are less differentiated in Saccharomyces uvarum, but because of metabolic constraints on galactose metabolism, having a secondary co-repressor is essential to maintaining that higher activity. Okay, so first let's focus on GAL1 and GAL3. This is the enzyme and the co-inducer for the network. In Saccharomyces cerevisiae, the co-inducer is dedicated. It has no enzymatic activity at all. However, if we go out, again, 10 million years, divergent, Saccharomyces uvarum, it turns out that we can show that the co-inducer, GAL3, is actually still has some enzymatic activity. So if we do a precise allelic replacement where we go in to Saccharomyces cerevisiae and put in the co-inducer sequence for uvarum, we actually can complement that enzymatic activity, and so in this case, uvarum GAL3 still has ancestral galactokinase activity. Similarly, we can look at the co-induction activity, and we see that the GAL1, the enzyme, is actually a better co-inducer than the cerevisiae GAL1, and that can be seen in a GAL3 knockout phenotype. So the cerevisiae phenotype, here's wild type in red, and a mutant for the co-inducer in orange, and what you see is it takes it 3 days to figure out that there's any galactose around and turn on the network. In contrast, if you make that same knockout in uvarum, here in blue, and then in green, there's only a couple hours delay. And so that's because uvarum still has a GAL1, just like cerevisiae, but its activity at co-induction is higher. And we can show, again by a precise allele swap, that that is due to differences in the promoter sequence. And so if we just take the promoter from Saccharomyces cerevisiae and put it into uvarum in place, what we see is we get that long-term adaptation phenotype back. When we put in the cerevisiae promoter, because it's weaker, it takes it longer in the absence of co-inducer to figure out that there's galactose around. So we can also look at just those allele swaps, and we can show that in general, the cerevisiae GAL1, both at the coding sequence, here's the coding sequence swap, and the promoter sequence in galactose has lower activity, and so both have changed, and in both cases it's a cerevisiae activity that's lower. We can do the reciprocal swap and show that when we put the uvarum sequences in the cerevisiae, the activity is actually higher. Okay, so what about the second copy of GAL80? And this is maybe more interesting because in this case, this is a gene, GAL80B, that is not present in Saccharomyces cerevisiae. First off, to cut a little bit to the chase, Saccharomyces uvarum GAL80 has a similar function to cerevisiae GAL80. It is the main co-repressor that's responsible for keeping the gene network off. So what about GAL80B? What is it doing? So it turns out that GAL80B turns on very strongly only in the presence of galactose. And so we can see in non-inducing conditions, GAL80 is the main protein that's expressed, but GAL80B comes on and induces over 100-fold in the presence of galactose. And it's doing this in order to keep the network from over-inducing. And so when we do an experiment where we actually look at the GAL80 knockout in the presence of both glucose and galactose, as a fitness defect because the network over-induces. So if we remove both GAL80s from uvarum, both GAL80 and GAL80B, perhaps not surprisingly, we get a constitutive expression phenotype. And so this is a reporter gene for GAL1, that enzyme, and this is just showing that if we knock out either gene by itself, we get maybe a little bit of induction, especially with the GAL80 mutation, but if we knock out both co-repressors, we get a whopping increase in gene expression. Okay, so what is the phenotype of overexpressing the GAL network? So it turns out in cerevisiae, this is well known. If you remove the co-repressor, you actually end up growing much faster. And in fact, if you switch between galactose and glucose, knocking out the co-repressor is a good thing. It allows you to induce and grow faster. And so that's shown here in orange. There's the wild type and there's the mutant in cerevisiae. You grow faster. In uvarum, we see exactly the opposite. Here's the wild type, and the knockout undergoes a massive temporary growth arrest. And so now if you are skilled at phenotyping yeasts instead of vertebrates, as we discussed earlier, you'll see, if you look really closely, uvarum, that knockout, that co-repressor knockout, is actually doing better early on in the culture. And indeed, we can actually look and we can see it's actually making more ethanol, so we do believe it's actually metabolizing quite a bit early on. And if you have really keen eyes, you can actually see that cerevisiae is actually growing, you can actually see that cerevisiae is also growing a little bit faster early on, undergoes a very brief arrest, and then increases. And this just shows that that's reproducible across multiple experiments. Okay, so what I'm arguing is that when you remove the co-repressors in uvarum, it's actually growing very fast early on and then overloading metabolism. And so there's been recent work done by Maitreya Dunham, Amy Cawdy, and more undergraduates than would fit, that have, as well as a follow-up study by Rachel Brim's lab, that have shown that in general, it's not just the data I showed you on GAL1, in general, the uvarum network is more active, and so that's consistent with this idea that it might be overloaded. So what does metabolic overload look like in cerevisiae? So this is a recent study by Bastusink's group, and what they showed is that if you provide glucose to Saccharomyces cerevisiae and force it to start growing, it can enter an imbalanced state if it consumes glucose too fast. And what happens is, this is because of the investment steps of metabolism, end up consuming ATP and depleting phosphate. And so you actually end up accumulating fructose bisphosphate and depleting phosphates. And so in Saccharomyces cerevisiae, this is offset by something called the trehalose cycle that actually, in the early phase of glycolysis, takes over a quarter of the glucose and actually runs it through this fetal cycle that regenerates phosphate. It can also regenerate phosphate by glycerol production. And so if this doesn't happen, that's where you get the fructose bisphosphate overproduction. So we did metabolomics by LC-MS with Josh Kuhn's lab, and so what we see is in our uvarum co-repression mutants, we see accumulation of metabolites, especially in upper glycolysis. We see before TGA, we see a 13-fold increase in fructose bisphosphate, and we see in particular trehalose 6-phosphate and glycerol 3-phosphate accumulating a whopping 80 to 225-fold. And so these networks are really stressed out and trying to deal with too much flux going through too quickly. So I've shown you evidence of that the GAL1 activity is higher. I've also shown you that the GAL3 enzymatic has enzymatic activity, unlike in Saccharomyces cerevisiae. I didn't have time to show you a couple other pieces of evidence that uvarum also has higher GAL network activity. It has a second transporter. PGM1 is also a direct target of GAL4 in Saccharomyces uvarum, and so this actually goes up in response to GALactose, unlike in cerevisiae. So what we see is the structural part of the gene network is geared to a higher activity. It has higher expression. And it is prone to fluxing too much GALactose through. The only thing that's offsetting that is having that second copy of the co-repressor. So we wanted to see if these two phenomena were related, if the co-repressor, having that secondary co-repressor, actually enables the higher activity. So to do that, we looked again at the double knockout of uvarum, and we now replaced the GAL1 coding sequence or GAL1 promoter. And when we do that, we see either both the promoter and the coding sequence just turning down that metabolism actually provides some protective effect. So what I've shown you is that 100 million years after the whole genome duplication that created GAL1 and GAL3, we still have functional differentiation going on among these ancient paralogs. This is having functional consequences that allows the uvarum network to have more activity, the cerevisiae network to be more tightly regulated, and indeed, in both of these cases, both GAL80 and GAL1 and 3, there are strong functional differences. I've also shown you that you can have metabolic overload if the control systems are bypassed. And finally, I've shown you that there's a connection between having that more active network in uvarum and the more robust repression system in order to keep it in check from over-inducing. And of course, this has been the high tempo of model organism research. It's been a great time, but I also want to highlight that we do need non-traditional model organisms as comparative systems. That temporary growth arrest phenotype, even though it is homologous and is present in cerevisiae, was missed by decades of previous research because it's much stronger in uvarum. And so finally, I want to thank Christina for doing all of this amazing research. This is a part of her dissertation. If she were here, she would be trying to meet with people doing molecular evolution in behavioral genetics and neurogenetics. And so if anyone is in that area, please contact me. And of course, she would also want to thank her lab members, many of whom have posters or workshop talks. Thank you.
Time for a question?
I was just wondering, like, how distant in sequence GAL1 and GAL3 are? Are they like very like how many amino acid substitution roughly?
What you mean? What percentage of amino acid identity?
Yeah, like are they still like very similar, or is it like a very large difference?
So genome-wide, it's about human chicken level of sequence divergence between uvarum and cerevisiae, but you're asking about the paralogs, right? I think that is closer to 70% sequence identity.
Thank you very much.